Healthy Smile, Happy Child initiative is very happy to have our two guest speakers today. They are local, but I think they will really amaze us with the work that they've been doing. I will start first maybe with the more recent person to finish their PhD. So Dr. Wasif Khan, so quite excited as of yesterday, convocated with his PhD from the University of Manitoba, and he was under the supervision of co-supervision of Dr. Chelikani and Dr. Who's now at Western University. So Wasif's research focused on computational and bioinformatics approaches to investigate the role of microbial, taste genetic, and social behavioral factors in early childhood caries. So it's really neat to see how those different realms are connecting now. And Dr. Khan is particularly interested in developing biostatistical and multi-omics models for large-scale data sets to identify the patterns in biological diseases. So Wasif, congratulations and thanks for joining us today. We're excited. Dr. Chelikani is a professor in the Department of Oral Biology and currently our Associate Dean for Research within the Dr. Gerald Nisnik College of Dentistry at U of M. And Prashen's research interests over the past 2 decades have focused on understanding taste biology in humans. His long-term goal is to understand taste protein-mediated host micro interactions and pathophysiology. And to achieve this goal, he has undertaken a broad range of research activities. One of his areas of research is focusing on analyzing the association between taste genetics, oral microbiome, and oral health using distinctions-based approaches in different cohorts with different populations. So I welcome both of them today. I'll turn it over to them. I'll mute myself. We will have good time for questions. And Prashen and Wasif, before you get started, let us know if you want to accept questions sort of as they come up, or if we hold them to the end. But absolutely welcome. Thank you so much, and over to both of you.
Okay, thank you, Dr. Shroth, for the kind introduction, and Daniel for organizing the Happy Smile, Happy Child series. And it's a pleasure to actually present today. This work, as Dr. Shroth mentioned, is part of Dr. Khan's PhD thesis, the newly minted Dr. Khan, right? So we are going to talk about what we have done. So this started way back, the study in 2017, and we are going to talk about these things. Before we get started, we would like to acknowledge the traditional territories acknowledgement. The University of Manitoba campuses and research space are located on the original lands of Anishinaabe, Inuvik, and Asinunuvik, Dakota, Oyate, Dene, and Inuit, and on the national homeland of the Red River Métis. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. Next slide, please. Yeah. Early childhood caries, most of you might know. And for those of you who are not familiar, it's defined as any caries experience in young children, less than 72 months or 6 years old in their primary dentition, also referred to as the milk teeth. And there's no one factor that causes these cavities in young kids. It's multifactorial, and predominantly until the past few years, S mutants, Streptococcus mutans, is known to be the key pathogen responsible, right? And it has been well studied for the past 2 decades or more, right? And it's endemic, I would say. Worldwide, we have close to 500 million young children affected by these. And by this ECC, it's 5 times more common than asthma, 7 times more prevalent than common allergies. So it's highly prevalent in certain communities within Manitoba. This is a chart a few years old, but I think the trend remains the same. You can see Winnipeg versus the rural versus urban divide, right? In Winnipeg, we have uh, comparatively less compared to the rural communities where ECC is much more prevalent, right? And also the newcomers, they also have sort of significantly higher compared to the general public. So moving on to the next slide. So the factors that affect these caries are multiple, right? This is a nice cartoon that was created by Dr. Khan using AI, I believe, right? So it shows both the risk factors and things that are protective, right? The risk factors includes bacteria, the known risk factors, right? And the dental plaque, retention of it, sugars, dietary consumption, right? We'll look at some of these in this talk. And the protective factors, saliva. When we talk of saliva, you're talking of circulation of saliva in the oral cavity, right? So there are certain conditions where if there is not enough saliva secreted, that also can lead to cavities and other oral disorders. Then we have fluoride and toothbrushing. So preventive strategies, right, which are protective in nature. And on top of these, we have many individual factors such as the microbiome. Microbiome, when we talk of this, people always think of just bacteria, right? Microbiome includes bacteria, fungi, viruses, phage or phage, right? So there are multiple things at play here, right? But it's very tough to study all of them, right? So bacteria is the highly studied one. In our studies, we also included fungi, and that is something Wasif is going to talk about, right? And then we also have genetics at play. When we talk of genetics, it can be genes associated with enamel formation, right? And also genes that are associated with diet, right? So the genes we sequenced in this study are mostly concerned with diet, taste preferences, right? And there are other factors at play such as family, socioeconomic status, health behaviors, and at the community level too, right? Social and physical environments can play or impact the development of ECC. So ECC is truly a multifactorial disease, right? Not one causative factor, right? Causation. We can't blame just S mutants as the causative factor, right? Next slide, please. Human taste perception. This is something people often overlook. They get confused between the different types of taste. Humans, as we know, they can sense 5 basic tastes, right? Basic taste sensations. And these are listed here. Sweet indicates energy-rich nutrients such as carbohydrates. Bitter allows sensing of natural toxins. Not all bitter compounds are toxins. Some are very healthy foods like green vegetable extracts, right? Those are bitter but very good for health. And then we have the salt, the taste of acids. Salt is the electrolyte balance, sodium salt, sodium chloride. Umami, that's the taste of amino acids or the meat broth, right? And on the right panel, you're looking at different uh, receptors that actually sense these basic taste sensations. For sweet and umami, we have 3 receptors. Depending, 3 receptors means 3 proteins that are present on the plasma membrane, right? And if you look at umami and sweet, the green is the common protein. That's the T1R3, right? And they differ only in the blue and orange. So for sweet to be sensed, you need both the orange and the green protein coming together, and the sugar binds on the extracellular side, that is outside, right? The surface of the cell. The same thing happens with umami, right? And with beta receptors, these don't have this huge N-terminal domain that is present in the other sweet and umami. But beta receptors, there are 25 bitter taste receptors in humans compared to those 3 for sweet and umami, which makes sense because if you look at those, the natural toxins, there are a lot of them, hundreds of them out there, right? So we need a bigger number of bitter taste receptors. That's why we have 25. We don't know what they do. Like they're present all over the body, not just in the oral cavity. Depending on what they are or where they are present, they are going to do multiple functions. Right? Salt and sour. Sour is more of for protons, right? And acids. So this is the schematic regarding the basic taste sensations. When people talk of hot, right, that's actually pain perception. It's not taste, basic taste sensation, right, or perception. So there's a difference between sensation and perception, right? So can you please move on to the next slide, please? Yeah, thank you. So human taste coding. This is something that a lot of people get it wrong even now. The traditional hypothesis was the tongue map hypothesis. According to the tongue map, different regions of the tongue code for different taste sensations, right? But that's no longer relevant or not scientifically proven. So it's not a valid concept. So then it brings up the questions for what are the mechanisms of taste coding in humans. There are 2 models out there, label line model and a cross fiber model, right? And at the bottom panel, you're looking at those 2 models. This is a human taste bud, right? And if you look at the color coding, right, each taste receptor cell. So these taste buds are like rose-shaped structures, right? The rose-shaped structure has multiple cells, and those cells are referred to as taste receptor cell or TRC. And according to the label line model, each TRC codes for a specific taste, right? For the across fiber model, each TRC or the taste receptor cell has all the taste proteins, right? All the taste receptors. That's the fundamental difference between these two. And there are studies still going on to prove which one is better, right? So there's no consensus yet on which is the right model. Yeah, next slide, please. Moving on to our own studies, right? So we were looking at the connection between the oral microbiome, genetics, specifically the chase genetics, and oral health. Depending on the cohort, for example, we are looking at a cohort in adults with rheumatoid arthritis, how genetics, taste genetics influence microbiome and oral health. In that population, we are also looking at periodontal and peri-implant diseases in adults, right? And coming to the last one here, which is the biggest one, right, which we are going to discuss in this current lecture, early childhood caries. So the study started in 2017, and it was a pilot study. We looked at 80 kids, 40, it's a case control study, 80 kids initially, 40 with ECC, and 40 caries free, right? And once we got the CIHR funding, then we started the big study, which started in 2018 and it ended in 2023, right? So it took us more than 7 years because of COVID and other things. It slightly delayed, but we were able to recruit 550 kids, right? So this is the largest Canadian case control study so far that involves ECC. And Dr. Khan is going to discuss the results going forward. Thank you.
Yeah. Hello, everyone. Yeah, before going to our main research using on this topic, I just want to briefly mention that. So what is the connection between taste genetics and ECC? So previous studies have identified a few genes where they realize that the variants in those genes lead to dietary preferences. And initially it was assumed that due to those dietary preferences, maybe certain population or certain individuals have a particular diet, and because of that diet, microbial shift takes place, and maybe that is the cause of caries. But then there is another model, and according to this model, some of the molecules which are released by those microorganisms, such as AHL, And these molecules can be sensed by taste receptors. And in turn, these taste receptors can start a downstream signaling process, which can produce some antimicrobial molecules. And those molecules can inhibit or impact the growth of the same bacteria, or maybe the other bacteria or other microorganisms. And according to these models, it's not that the only dietary preference is the main cause of these variants and ECC, but there could be a direct relation between the microorganisms and taste receptors. And keeping that in mind, so we first started that what are some of the research gaps in the field of ECC that we should address. So first, we realized that most of the previous studies were underpowered because they did not have enough sample size. And on top of that, they were largely focused on bacteria. So it's really important to include fungi as a microorganisms when checking their relation with early childhood caries. The second reason was that we did not have enough information about socio-demographic variables and their association with ECC in particularly in Manitoba, because this study was focused on Manitoban population. Third, the clear indication of how taste genetics is related to ECC is not very clear, or was not very clear. We try to address some of those gaps. And fourth one was if there is any connection between taste genetics and microbiome. So this part was also not very well researched previously. So considering those research gaps, we formalized particular aims. So our first aim was first to investigate the association of various socioeconomic and behavioral factors in ECC. Second one is to see the microbial differences. And while doing that, we did not only check the microbial differences between caries-free and ECC conditions, but we also check the interkingdom interactions between fungal microorganisms and bacteria. Then we tried to check the association between taste genetic variants and ECC directly. And in the fourth part, we combined all these data sets together. So sociodemographic data, taste genetic variants data, and also microbiome data, which included both bacteria and fungi. And as a last step, we wanted to see like if we can check the influence of these genetic variants, which are present in taste genes, if there is an influence on oral microbiome, and particularly on the microorganisms which are involved in ECC. So to complete this research, or to do this research, we recruited 538 participants, which included both caries-free children, about 226, and there were 312 children with ECC. From these participants, questionnaire data was filled by either their parents or caregivers, which had information related to their food habits and toothbrushing habits, and many other questions which involved the information regarding rural-urban status, socioeconomic factor index. I'll explain that one little later. So some of the information were collected from the parents, and from the participants, two types of samples were collected. So first sample was dental plaque sample, and the dental plaque sample was used for microbiome sequencing specifically. The other sample was collected from buccal swab from the inner cheek, and this sample was used for the sequencing of 55 taste and taste-related genes. After this sequencing, the data was processed to obtain the microbial abundance data sets, both for bacteria and fungi. And also, we found the variants in taste and taste-related genes, which were included in this study. And initially, the individual data analysis was performed between microbiome data sets and ECC, taste genetics data and ECC, and later these two data sets were combined together for a holistic and combined analysis. And while doing so, we keep several factors as covariates or confounders. And these factors were age, sex, socioeconomic factor index, rural-urban status, and principal components. And we'll see when we go through the results, we'll see why these factors are important to be included as covariates in all the subsequent analysis. So the first result here is the result for the ECC association with socioeconomic behavioral variables. And this analysis was done in two ways. One is a univariate approach, where we check the association of each variable with ECC one by one. And one is a multivariable approach, which is a statistical approach where we combine all the variables together and then we check their association with ECC. If we focus on this part, which is multivariable result, so here on x-axis we have estimate, means the extent of their association with ECC for each variable, which are given on y-axis. If the variable is colored with red, it means that there is a significant association. The black variables are those where we did not see a significant association. If we look at the first one is child dental health, we see that that is strongly associated with ECC, and which is kind of obvious. Second one is ECOHIS score, and this score is Early Childhood Oral Health Impact Scale. So, and we can see that there is some correlation between this oral health impact scale and ECC. Then if we especially focus on SEFI, which is Socioeconomic Factor Index score, and we realize that this is also significantly associated. Looking at vaginal birth, so in our study, we found that vaginal birth was associated with early childhood caries. And the reason for including vaginal birth as a variable, because previous studies realized that the mode of birth has an impact on oral microbiome. And the reason for including this here is that if it modulates oral microbiome, that maybe the shift in oral microbial composition may lead to early childhood caries. And that's why it was included here, and we see a positive association. Age was also found to be positively associated. It means that as age progresses, we see that there will be more chances of getting or developing early childhood caries. And the next one is bedtime snacking habit. So children who have a tendency of using snacks at bedtime, they were found to be significantly associated with ECC. And then the last variable, which was very significant and negatively correlated, was urban status. So it means that children who live in rural communities are more prone to early childhood caries. We did not see any association of NutriStep score. So NutriStep score is a nutrition screening tool for every preschooler. And we did not see any association of this score and early childhood caries. So while we assume that specific nutrition or the food habits can lead to early childhood caries, but when a total combined score was taken, it did not came as a significant variable in this study. And among all these variables, we were highly interested in these two, which is socioeconomic factor index score and urban status, because we realized that these variables are highly important to keep in mind while dentists or practitioners do their practice, that these scores are very important, and the people who fall in those under those variables may be more prone to ECC. And then now I'm moving on to the microbial abundance part, so that how microbes are associated with ECC. And before going to that one, I want to explain that as in the field, we call this microbial abundance table as OTU table. And there is a reason, because OTU stands for Operational Taxonomic Unit, and where taxonomic units or levels could be anything from kingdom through species. Most of our analysis were limited at species and genus level, and because we've realized that they are more biologically relevant to see how species or at genus level, how these microbial abundances are modulated. And this is a typical example of OTU table or microbial abundance table, where we have samples in columns and individual species are given in rows, and their abundance can be checked here. for any sample for a particular species. And when we have samples from two different conditions, for example, in our case, from ECC and caries-free conditions, we can now use this table to analyze the data to see what are the microbial differences or microbial shifts between the two conditions. So here is the result for the microbial shift or microbial profile changes in ECC and caries-free. So all these red bars show that which of the microbes or microbial species or genus are highly prevalent in ECC, and their extent is given on this bar. And on these blue bars shows those species and those genus which are highly associated with caries-free, or we can also say that they were more depleted in ECC conditions. If we focus on top few species, I'll just zoom in this area. So if we focus on these species, the top one is Streptococcus mutans, as also mentioned by Dr. Chelikani, which is always considered as the topmost factor or topmost species behind early childhood caries. But interestingly, the second one is not a bacterial species or bacterial genus. The second one is a fungal genus Candida. So first one is a bacterial species. Second one is a fungal genus Candida. And the next two are also related to Candida genus. They're the two species of Candida, Candida dubliniensis and Candida albicans. Similarly, we can also see what are other bacteria involved in ECC. If we focus on caries-free conditions, so we can see the topmost species which are present in caries-free conditions are Fusobacterium periodonticum, Neisseria oralis, and similarly other species. So while doing this research, we also find something very interesting when we were going through how if these species may have any interactions among each other. And we realized that one of the very interesting interaction was identified between Candida dubliniensis and the bacteria Neisseria bacilliformis that we will see on the next slide. And I know this is a slightly busy slide. So what we are trying to show here is that we have captured all the bacterial or interkingdom interactions together. But the one I mentioned on the previous slide is given here, which is highlighted here. So here we can see that the species Candida dubliniensis is highly correlated with bacteria Neisseria bacilliformis. And this aqua color shows that this interaction was not present in caries-free condition, but this interaction changes to a positive interaction in ECC. So similarly, again, we can check all other interactions, but because of the limitation of the time, I'm just restricting it to the interesting one, which is between a bacteria and fungi. And while reading these kind of interactions, I want to highlight, which is a very important thing, that these kind of network plots do not show biological interactions, because these plots are captured based on the microbial abundances. So if the abundance of one bacteria, one species goes up together with other species, it will be shown as interlinked together on this graph. Now, because it's a long presentation, I would like to summarize some of the key results that we identified so far. So first, that we identified that socioeconomic conditions and rural-urban status were highly associated with early childhood caries. We identified ECC biomarkers. So we already know Streptococcus mutans, but fungal species Candida dubliniensis was one of the biomarkers for ECC. And we also identified interkingdom association between between Neisseria bacilliformis and Candida dubliniensis. And it is also important to discuss some of the limitations that's the point what I wanted to mention. These socioeconomic factor index scores are not based on individuals, but rather they were collected based on the postal codes. So this can be considered as one of the limitations, because these scores are not individualistic in nature. And second, again, which you may have heard, that we have to take these associations very carefully, because these associations do not mean causation. And there are different types of studies to establish to establish causation between microbial abundances and ECC or any other disease. Now we are going to the third part where we integrated taste genetics in this research. First, I wanted to give you an example of how the data looks like. So we saw an example of how the microbial data looks like. So this is a different example of how a genotype file looks like. So again, we have different samples from different individuals, which can be labeled as 1, 10, 11. And then in rows, we have the chromosomal positions, reference allele. Reference allele is based on human genome version 38, which is the most latest one. And then the alternate allele, that if there is a variant, that how it changes to a different nucleotide. Gene information, and then here these entries could be 0, 1, and 2, where 0 means there is no change. The sample is homozygous for reference allele. It means that, because there are 2 alleles, so it means both the alleles are same as reference. Where 1 means heterozygous, it means that one of the allele will be as reference, but other allele will be changed to a different one. And 2 means this particular variant would be homozygous for alternate allele. It means that both the alleles will be changed. So, and that is labeled as 2. And this is how we capture the genetic variants among all the individuals or the participants in this study. So this is the slide related to those 55 genes, which we selected as candidate genes. Again, as it was mentioned, that there are 25 bitter taste receptors, and so these are the name of the genes. So 25 bitter taste receptor genes, and then 3 of those genes which are involved in sweet and umami taste, and then salt taste, sour taste, fatty acid receptors, carbonic anhydrase genes. And then some of the genes are those which are not directly involved in taste, sensation, or perception, but they are involved in downstream signaling of the signal pathway. So in total, we started with 538 samples. We selected 55 candidate genes. And among those 55 taste genes, we identified 700 variants. So in all those participants in 55 genes, total number of variants were 700. But among those 700 variants, only 3 variants were found to be significantly associated with ECC. If we focus on the first variant is in the salt taste gene, SCNN1D, and it was a missense variant. It means that this variant changes the amino acid. So instead of original or reference amino acid, there will be a different amino acid. The second variant was found to be in bitter taste receptor gene, and it was a conservative in-frame deletion. It means the deletion was related to 3 nucleotides. So while it deletes 1 amino acid, which is based on 3 nucleotides, but overall protein, the remaining protein remains the same. And the third variant was in phospholipase beta gene, and this gene is involved in downstream signaling of bitter taste, umami, and sweet taste. And this variant was found in splice region variant, so not exactly in the coding region, but in between regions, but it changes how gene splicing will take place. So we found that these 3 variants were significantly associated with ECC. So at this point, we have microbiome data, genetic variants data, and socioeconomic and other behavioral data. And we tested their association with ECC individually. But then the question was that then how to know that which variables are more important than the others when we combine all the data sets together. And because the nature of these data are very different from each other, so to combine them, we relied a random forest model, which is a type of machine learning model. And when we combined these 3 data types and we checked it for their ECC classification between ECC samples and caries-free samples, we found that their performance was quite high, which was AUROC value 0.95, where the maximum value is 1 and the lowest value can be considered as 0.5. So on this scale, this value can be considered as very high. It means that the random forest model was able to capture the patterns between ECC and caries-free conditions very well. When we checked for the variable importance from this machine learning model, again, the bacterial species Streptococcus mutans came as the first candidate. The second candidate was rural and urban status. And again, this again highlights that just if the samples are from rural or urban status, it really have a large impact on ECC outcome. And more important than that was we were never expecting a genetic variant to be so high on this list, because ECC is considered to be a disease of microbial origin. But the involvement of genetic factor at third rank was very significant. And this was even higher than fungal species, Candida dubliniensis, and some other bacterial species. And that shows that genetic variants are highly important in ECC risk prediction. As a next step, as we said, that we wanted to see if those genetic variants, which are given on x-axis, can also modulate some of the microbial species. It means that if they have any influence of microbial abundance. So we checked the association of the three variants, which we found to be associated with ECC, with all the species in the oral cavity. And here we have the results for only those species where these associations between variants and microbial species are significant. And their associations are given by these colors, where the brown color shows positive association between these two factors, and green means negative association. And these asterisk shows the significance level. So more the asterisks are there, it means that the higher the significance or higher the association is. And among them, we found that 5 of these associations were significant even after false discovery rate adjustment. And those species are here. So now the obvious question would be that are any of these species the same as which are associated with early childhood caries or caries free? So which we saw on this graph, these red and blue bars. But we did not see any overlap between these species, which were ECC associated or caries free associated, and these species. So based on that, what we conclude that although these variants influence some of the microbial species in the oral cavity, but not exactly the one which are ECC associated. And then we use a different model to specifically check the pathway, whether these variants directly influence ECC status or they influence bacterial species, and that species in turn cause ECC. So we tried it for one highly associated with ECC, Streptococcus mutans, and the other species which is highly associated with caries-free condition. And when we check the pathway, we realize that if we focus on this beta value, I'll just zoom it in. If we focus on beta value, we realize that the beta value of this pathway, direct pathway, is more than this pathway. It means the influence of these variants on ECC is more than this pathway. It does not go through the species. Although this pathway is also somewhat significant, where p-value is very, very less, but there may be other factors involved, not the direct influence on bacterial species. So again, the conclusion remains the same, that while these variants influence microbial species, but not exactly the ECC associated one. So in summary, that we found that some of the genetic variants in bitter taste receptor, sodium ion channel, and phospholipase beta gene were associated with ECC. And then in multi-omics data analysis, we found that genetic factors also appear in among the top features. And then genetic variants do have some influence on oral microbiome. Again, talking about some of the limitations, that most of the genetic studies should also have ancestry information included while doing the analysis. But because we did not have this information, we could not do an ancestry stratified analysis. So this could be considered as one of the limitations. And other limitation is that we only considered taste-related genes. So we are not doing genome-wide association studies, but this study was limited to 55 genes, which we consider to be associated with different tastes. Now, I'm going through the overall significance of this research that we identified the role of a fungi, especially Candida dubliniensis, and its potential association with the bacteria, which was Neisseria bacilliformis. We also realized that there is a significant association between socioeconomic factor index score and rural-urban status, and this can help policymakers regarding oral health. Our study establishes an association between taste genetic variants and ECC. And then in addition to ECC, it also checks the association between genetic variants and oral microbiome. And in the end, we can say that this study advances the understanding of ECC by integrating and comparing the relative importance of different variables, different data types, genetics, microbial, and social determinants. At this stage, it's also important to see what could be some of the future directions. So one and the very important one could be some longitudinal analysis on microbiome data to establish the causality. So as I mentioned, our research, which was based on case control analysis, cannot establish a causality between microbial abundance and ECC, while a longitudinal analysis can do that. Second one is, although our sample size is considered as a very big one, while considering microbiome data, so 500 data points for microbiome data is a huge one, but that is still limited for genetic association analysis, so which still requires a bigger sample size. Then, as I mentioned, this was a limitation that we have to do a stratified analysis for ancestry to exactly check the genetic association between any outcome. And while doing so, socioeconomic conditions should also be considered if the study is related to ECC. Then we just check the variants and their association with microbial abundance, but in the future, the genetic gene expression level can be compared with microbial abundances and also with microbial metabolite presence, so amount of them. So, and this requires RNA transcriptomic data, microbiome sequencing data, and also metabolite sequencing and other high throughput sequencing. And the most important one in them is to always check or investigate the biological mechanism behind those interactions or associations. So once we find something, we have to go back to the lab and then thoroughly check it if what could be the biological mechanism behind those associations. And with this, so all the results that I have mentioned or we discussed in this presentations are given on these two publications, which were recently published, and Dr. Chelikani is the corresponding author. So if you go to Dr. Chelikani's website, you can find these papers easily, and you can see some other results also, which we could not discuss due to the limitation of the time. And with this, this is the end of the presentation. I would ask Dr. Chelikani to see if I forgot to mention something important and if he wants to highlight that. So Dr. Chelikani?
Oh, yeah. So, it's good. And we would like to thank the participants and caregivers who actually were very patient throughout the study. As we mentioned, it's the largest study in Canada so far, even though it's restricted to Manitoba population, but it is the largest study. So we sincerely thank all those participants and the caregivers, parents who took part in the study, and also our funding agencies. Thank you.